Hello everyone. This is 3 p.m. and most of the people start daydreaming during this time, but let's challenge this thing by staying engaged. A very good afternoon to everyone. I am Bushra, your botany faculty, and today I'm going to discuss questions from this paper AIATS Plus Phase 2 Mock Test 2, which was held on 26th of January, and specifically those questions which are mistaken by many students in this paper. So in this lot, I am going to discuss questions basically from these chapters. Two chapters are from 12th. First one is molecular, next one is biodiversity and conservation. Molecular basis of inheritance is one of the most favorite chapter for NEET point of view. Now, next three chapters are from class 11, biological classification, anatomy of flowering plants, and plant growth and development. Okay. So now let's start questions particularly from these topics. Now let's start with the very first question, that is question number 126, which says which of the following activities can be performed by enzyme DNA dependent RNA polymerase in a prokaryote. As you can see that this question is from molecular basis of inheritance. Here in the question itself, it is asking about the enzyme. From the enzyme, you can see that it is telling DNA-dependent RNA polymerase. That means RNA is polymerizing by depending on DNA. Now tell me very fast that what this process is known as. Come on students, tell me what this process known as, where DNA transcribe, or we can say transcript, answer itself in the statement that this is known as transcription, that means formation of RNA from DNA. So this enzyme is involved during the transcription process and in prokaryote. Okay, now let's start with transcription in prokaryote, as the enzyme here is DNA dependent RNA polymerase. First, let's see what are its properties. It is the only enzyme which is involved in the transcription of prokaryote, and it catalyzes the transcription of all types of RNA in prokaryotes. For example, mRNA, rRNA, and tRNA. And if we talk about the polarity, कि ये किस side catalyze करता है, so it catalyzes from 5' to 3', right? And one more property of this enzyme is that it facilitates the unwinding of DNA double helix. Remember that it is doing unwinding of DNA double helix during the process of transcription. This type of question is also asked in the NEET exam, that which enzyme is involved during the unwinding of DNA double helix during the transcription. Here it is DNA dependent RNA polymerase, but in DNA replication it is helicase. Okay. So remember this point very clearly. And the most important thing is that how this enzyme look like. It basically consists of 5 subunits. What are those subunits? 2 are alpha, 1 is beta, another is beta dash, and the 5th one is omega. When these subunits are present, then the enzyme is known as core enzyme. Right? So these are the properties of this enzyme. Now, if we look into the transcription of prokaryotes, it is basically divided into 3 steps. First one is initiation, next is elongation, and the third one is termination. So, how this enzyme actually does the work? When this core enzyme get attached, or we can say get provided the specificity of initiation from sigma factor, which basically direct it towards the promoter region. It takes this enzyme, the core enzyme of DNA-dependent RNA polymerase, to the transcript, from where the initiation of transcription will occur. Right? Now, after the initiation, this sigma factor gets released. And then what we are left with? Only core enzyme. So this enzyme will facilitate the process of elongation. Once the elongation of RNA is completed, then one more factor will come, which is known as rho factor. This is the sign for rho, and then it will terminate the process. One more thing, once the core enzyme get attached, or when sigma factor get attached to this core enzyme, then it is known as holoenzyme. So this holoenzyme has the capability to initiate the process from the promoter region. But when it is not along with any of the factor, then it has the capability for elongation. It is like a very important thing to understand that alone core enzyme can perform the role of elongation, but once it get attached to the sigma factor, it will initiate the process, and when it will attach to the rho factor, it will terminate the process. This type of questions are also asked in the NEET exam. So you have to be very clear that which factor will initiate and which factor will terminate. Now let's see the very beautiful diagram from the NCERT. Here the initiation of the process is shown, where you can see that RNA polymerase is getting attached with the sigma factor and how it is initiating the process by attaching this RNA polymerase to the promoter region from where the initiation of the transcription will start. Later the sigma factor gets removed, and alone this core enzyme is once opens the DNA double helix structure and then initiate the process of elongation. That's why we say that the core enzyme has the capability for the elongation process, right? Now next one is, once the complete process is done, then this RNA polymerase gets removed along with the rho factor, and there will be release of nascent RNA. So this is how actually the process look like. Along with that, one important, or we can say beautiful line is written in NCERT, which says, an intriguing question is that how is the RNA polymerase able to catalyze all the three steps? As I've told you that this initiation is done by sigma factor, elongation is done by core enzyme. I hope you are able to see this red color. And this termination is facilitated when it gets attached to the rho factor. And the next line it is written, it associates transiently with initiator factor, initiation factor and termination factor to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate, right? So this is the whole concept behind the RNA polymerase. Now let's get back to the question. So these are the activities which are shown by DNA dependent RNA polymerase. First activity is polymerization of deoxyribonucleotide. Here the formation of RNA is taking place, that means RNA is taking place, ribonucleotide. So this is incorrect. So from this I can rule out the option which will contain A. That means option number 1. Okay, now next one is unwinding of DNA helix. As I have told you that one of its function is to facilitate the opening of DNA double helix, right? Now tell me the enzyme which is involved in the unwinding of DNA double helix during the DNA replication. Please write down in the comment box. Okay, in the meanwhile, let's see the next option, next activity. Next is synthesis in 5' to 3' direction. Yes, it is also shown by this enzyme. So हमको वो वाला option choose करना है जिसमें B and C दोनों हो. That means option 4. Now if we see the next option which says joining of Okazaki fragments, these are formed during the DNA replication, not during the transcription. So this option is incorrect. Next one is ribosomal RNA synthesis, as this is the only enzyme which catalyzes all the types of RNA in prokaryotes. So this is also one of its features. So along the 5 features, 3 are true, that is B, C and E, which is present in option 4. So from here, I can directly hit the correct answer, right? Now let's move to the next question. Again, this is from the same chapter, molecular basis of inheritance. I don't know why people, suffer a lot of problem from this chapter. Actually, this chapter is like very technical or, samajhne wala hai, and it is really interesting. By the way, let's see the question. Read the following statements of assertion and reason and choose the correct option. Assertion says DNA replication in E. coli initiates randomly at any place in DNA, and reason says E. coli DNA lacks origin of replication site. So the underlying concept behind it, replication of DNA. Let's understand it. It is basically कोई भी process को समझने के लिए let's break them into steps. Now let's start with the first step, which is origin of replication. What do you understand by origin of replication? It is basically a small sequence from where the replication will be started. And if we talk about the prokaryotes and eukaryotes, in prokaryotes, there are more than one origin of replication. In eukaryotes, single origin of replication is present. Now, if we see the next step, which is activation of deoxyribonucleotide. Students, make this session as engaging as possible so that I can seep my thoughts into your brain and make you understand what these things are. Okay, so here activation of deoxyribonucleotide will occur where the action of phosphorylase enzyme will take place. That means addition of phosphate group, and it will lead to the formation of nucleoside triphosphate. That means there will be addition of phosphate group in them. Now this will act as a substrate for the process of replication. Along with that, it will provide energy to the process. How it is actually providing energy? As the phosphate groups are attached to it, once they are broken down, then it will release the energy which will be utilized in the process of replication. Now, let's see the next step, which is, let's mark it as 1, 2, and third step, which is unwinding of DNA double helix. Just in the previous question, I have told you about the transcription, that which enzyme is involved during the, which enzyme is actually involved for the unwinding of double helix during transcription, that is RNA polymerase. Now, if we talk about the replication process, here enzyme involved is helicase. Please remember these things, okay? So this enzyme basically unwind the DNA double helix and make it like a bubble, like this. Right? Now, if you can see that these two strands are far apart from each other. So to stabilize them, there is one protein known as SSB protein, single strand binding proteins, which are required to stabilize those single strands which are formed due to this enzyme. अब जैसे कि enzyme आया and it unwind the DNA double helix, it created the tension here. These are also supercoiled. यहाँ पे loosen up हुआ, that's why it created a tension here, which is further released by another enzyme known as topoisomerase. If we talk about prokaryotic organism, this function of this enzyme is carried out by DNA guidase. If we talk about prokaryotes. Okay, so don't get confused between the topoisomerase, DNA guidase and helicase. All these enzymes are actually doing the functions in the third step. Okay, now if we see the fourth step, which is, I am writing here, which is formation of primer. Why primer is required? For initiating any type of process, one stepping stone is required, which will be given by this enzyme, which is primase, and it will form primer, which is the small stretch of RNA. Right, so this will be basically the stepping stone for DNA replication. And this will fall off once the initiation of DNA replication will get started. Enzyme involved here is primase. Okay, now let's see the next slide here, how it actually look like. जो यहाँ पे हमने देखा था this bubble, if we study it like this, then it will form a replication fork. It is actually forming the replication fork where unwinding of DNA double helix takes place. Right? So यहाँ से ये खुला हुआ है and here newly synthesized strands are forming. Now let's see the polarity. Fifth step, that is polymerization, where the role of enzyme, which is very important, DNA dependent DNA polymerase, a very important enzyme for the process of DNA replication. Here you can see in the name itself that it is polymerizing the DNA by depending on the DNA template itself. Okay, so here replication fork is formed. Now let's see the polarity. 3' to 5' here DNA is synthesizing in one go, because the formation, or we can say the catalysis capacity of this enzyme is from 5' to 3'. That means it will attach to the 3' of the template strand and it will start polymerizing the DNA towards 5'. Right? So here 5' to 3' new strand gets synthesized, as these are complementary to each other. This is continuous strand. Right? There is no break. Now, if we see the next strand which have the reverse polarity, that is 3' is towards here and 5' is here. But how DNA dependent DNA polymerase work? It work from 5' to 3'. That's why jitna open hoga DNA double helix, utna hi stretch of DNA it will form and then it will get fall off. That's why discontinuous strands of DNA are formed in the next strand. These are discontinuous strands, which is also known as Okazaki fragments. In the previous question, we have seen this as an option. And these fragments are later joined by another enzyme known as DNA ligase. As the name itself suggests, ligase yani ki jodna. So here this enzyme will come into role and joined all the Okazaki fragments later. So this is how the replication of DNA takes place in prokaryotes. Properties of DNA-dependent DNA polymerase, if we talk about the prokaryote, for example, E. coli, the complete process of replication will take place in 18 minutes. This is one of the PYQ, and it has the capacity to polymerize base pair, 2000 base pair per second. Again, this information is given in NCERT plus NEET has also asked this question. Now let's get back to the question, which is assertion reason. The first statement is DNA replication in E. coli initiates randomly at any place. So no, this is not true, because origin of replication is required for initiation of replication. So first statement is incorrect. So from this only I can hit the correct answer. Right? So this is the way, or you can say the way how we can solve these type of question. Assertion reason dekhte we are like, oh my god, such a difficult question. But this is not the case every time. If we see the reason which says E. coli DNA lacks origin of replication site, so this statement is also false because it does contain. Right? So this is how we solve the question. Now let's move to the next question, which is 113. And this question is from the chapter Anatomy of Flowering Plants. Okay, I will not use the purple color. Okay, thank you so much. Now let's see next question, which is from anatomy, which says collenchyma differs from parenchyma as the. So as in the question, two simple tissues are given, collenchyma and parenchyma. These are simple permanent tissue. Okay, what are simple permanent tissues? Those tissues are composed of cells which are structurally and functionally similar to each other, and they perform the coordinated function, and they destined to form a same function. That's how they form the simple permanent tissues. These are of three types. First picture, very beautiful picture, is of parenchyma. Next image is of collenchyma. This picture is of sclerenchyma. These two are basically types of sclerenchyma. This one is fiber, and these are generally found in the fruit walls, sclerites. Now, if we see the feature, the type of cell, that how cells actually are in these type of tissues. Okay, so one is asking कि AR गलत हो जाते हैं हमेशा. So here for assertion and reason question, we have to always link the statements. If in the next paper of if I see in the upcoming questions some AR is given which is quite difficult, or one or two, then I will explain it properly in that question. Okay, now let's get back to this concept where, so the type of cells which are present in these type of tissues. At maturity, the protoplasm is present in the cells of parenchyma. That's why the cells are living. It is composed of living cells. If we talk about collenchyma, here protoplasm is also present when they are matured, so they are also living. But if we talk about the sclerenchyma, at maturity, cell lose their protoplasm. That means those are dead cells. So these tissues are basically composed of dead cells. If we talk about shape, they are generally isodiametric. Isodiametric, that means they are oval or round in shape, of similar size basically. They may be in oval, polygonal shape also. If we talk about collenchyma, they are oval, polygonal or spherical shape. If we talk about sclerenchyma, they are narrow and elongated. Narrow and elongated. Now, if we talk about the cell wall. Cell wall in the parenchymatous tissue have the deposition of cellulose. If we talk about collenchyma, very interesting feature, seriously, that the cell wall of collenchyma have the deposition of cellulose along with hemicellulose and pectin, but they are not evenly distributed in the whole complete cell wall. They are deposited at the corners of the cell wall. So these red-like structure are basically deposition of these things, cellulose, hemicellulose and pectin, which help in providing mechanical strength to the growing parts of the plant, for example, petiole of the leaf. Okay, now if we talk about the sclerenchyma, so here the cell wall has the deposition of lignin, which provide them strength. Right? Now, if we see the functions, for functions, if we talk about parenchyma, so it performs the function of storage, secretion. If photosynthetic pigments are present, then it is known as sclerenchyma and perform the role of photosynthesis, right? And if buoyancy is provided for a plant, कुछ plants में buoyancy की ज़रूरत होती है, for example, aquatic plant, then air bladders are present in the parenchyma. Those are known as arenchyma, which provide buoyancy to the plant. If we talk about collenchyma, just now I've told you that due to deposition of these things at the corner, they provide mechanical strength to the growing parts of the plant, and they are present in the hypodermis of dicot stem. Right? One more feature which is very important for collenchyma, that they are absent in monocotyledonous plant. I am writing here absent in monocotyledonous plant. जैसे कि आप देखेंगे monocotyledonous stem के hypodermis में collenchyma नहीं sclerenchyma होती है. Okay? Now, if we talk about sclerenchyma, so here two types are given. Here fibers and this is sclerid. Fibers are found in the mature plant trunk or tree के trunk or stem में होते हैं, which provide them mechanical strength. Right? Lignin का deposition है, which make them strong and provide mechanical strength. If we talk about the sclerids, they are found in the pulp of fruit, for example, sapota. Sapota को हिंदी में क्या कहते हैं? Can anybody tell me what do you understand by sapota? आजकल उसका season भी चल रहा है. In a meanwhile, let's move further. फिर बताते हैं कि sapota actually क्या होता है. And they are also found in the walls of nut. Right? जो आप fruits खाते हैं उनमें हल्का सा gritty nature आता है ना, that is due to the presence of sclerids. So next time when you eat fruits, so remember that these are actually sclerids. Okay, so I think we are done with the functions. Okay, and these parenchymatous tissues are basically present in most part of the plants. सारा plant इसी से ही बना होता है, basically parenchymatous cells से. Right, so this is all the features between the simple permanent tissues, or we can differentiate it here. Now let's get back to the question, which is actually asking the difference between these two tissues. Collenchyma differs from parenchyma as the first option is former has cell wall thickened at the corners with cellulose, hemicellulose and pectin, as in the the session which I have conducted just a minute before, where I have told you that the very beautiful feature of collenchymatous tissue or cells which have the thickening at the corners of cellulose, hemicellulose and pectin, and yes, it is the differentiating feature between these two tissues. So actually this is the correct answer. Let's see the other options also. Later is living. Here both the tissues, the cells which are involved in the tissues are living. So this is not the differentiating feature. Third option is later is dead mechanical tissue. Later, that means parenchyma, but it is made up of living cells. So ये भी incorrect हो गया. And the fourth one is former can store food. This is also incorrect, right? So this is not the differentiating feature between them. So for the question, the correct answer is option 1. Now let's move ahead to the next question. That is question number 146, and again from the same chapter, that is anatomy of flowering plant. I think this is a type of Nidhi, yes, very good Nidhi. You know that sapota is chiku. Very good. Now, let's see the next question again from the same chapter. 146, which says how many of the given features is or are true for dicot stem. Now, let's understand how dicot stem and monocot stem actually look like and what are their differences. This is the image of dicot stem, or we can say TS of dicot stem. And this is the transverse section of monocot stem. TS is the short form for transverse section. Don't get confused that I have used some another term. Okay. So if we look into the picture, we can see they both have epidermis. It is common in them. So I will write here parenchymatous epidermis, right? Now, if we talk about the hypodermis, so in dicot stem, just previously in the question, while explaining the collenchyma, I have told you that hypodermis in dicot stem is collenchymatous. Feature is hypodermis. And if we talk about the monocot stem, in them hypodermis is sclerenchymatous. Right. Now, if we talk about endodermis, very beautiful feature of dicot stem of endodermis, where there is deposition of a starch grain in the endodermis. That's why it is also known as starch sheath. And this question is asked in the NEET exam also. So it is very important that which structure of dicot stem is also known as starch sheath, right? But if we talk about the monocot stem, endodermis is not present. In the monocot stem, I would like to tell you that cortex is not differentiated into further different layers. So endodermis is absent, pericycle is absent, pith is also absent. Now if we talk about next layer, which is pericycle, present in dicot stem but absent in monocot stem, which is sclerenchymatous. It have patches of sclerenchyma, right? And it is present in a semi-lunar shape, which is just beneath the endodermis and above the phloem, right? Now, next one is vascular bundles. If we talk about vascular bundle, I am writing VB for vascular bundles. So, in both of them, conjoint vascular bundles are present, right? But one differentiating feature that in dicot open type of vascular bundle is present. That means between xylem and phloem, cambium is present, as dicot stem will undergo secondary growth which required cambium. Okay? But in monocot stem, cambium is not present. That's why it is known as conjoint and closed. Okay, so this is the differentiating feature between the dicot and monocot stem. If we talk about the arrangement of vascular bundle, it is ring-like arrangement. It is also the characteristic feature for the dicot stem. And the vascular bundle you see here is actually scattered in the whole stem. So if we talk about the arrangement, it is different in both. And in scattered also you can see the vascular bundle which are large in size are actually present at the center, and the vascular bundle which are smaller in size are present towards the periphery of the stem. So this is how actually the vascular bundles are arranged in monocot and dicot. Now if we talk about next structure, which is pith. This is pith, but in monocot stem you cannot see pith because इसमें होता ही नहीं है. It is not present in monocot. Okay, and in dicot stem you can also see one more structure, medullary rays. These are also known as pith rays. The parenchymatous cells which are present in the pith are actually get extended in between the vascular bundles, these region, and they are known as medullary rays. Right, so these are the feature, or we can say the differentiating features between the dicot stem and monocot stem. Now let's get back to the question where it is asking about how many of the given features are actually true for dicot stem. So here we have to count the number of features which are true, which holds true for dicot stem. The first one is presence of sclerenchymatous hypodermis. So it is incorrect, as just now we have studied that it has sclerenchymatous. Okay. Now, next feature which says absence of water containing cavities in vascular bundle. So yes, it is true. I'm so sorry that I haven't mentioned it here. In monocot stem, vascular bundles have water containing cavities. These are its characteristic feature, this water containing cavities. If someone gave you TS of monocot stem, how will you identify? There will be presence of water containing cavities, but they are absent in dicot stem. Now let's see the question which says absence of water containing cavities. So yes, it is true for dicot stem. Third feature which says presence of conjoint and open vascular bundle. So yes, it is absolutely true, as dicot stem will undergo secondary growth which require the presence of cambium. That's why it is known as open. Now, this is also the correct feature. Next one is presence of sclerenchymatous pericycle. So yes, it is also true. Pericycle is present beneath the endodermis and above the phloem. Okay. Now, next feature is occurrence of endarked protoxylem. This feature is actually common in both monocot and dicot stem. Here I will explain this endarked protoxylem. What do you understand by that? If we talk about the arrangement of primary xylem, then protoxylem is actually present. Protoxylem is actually present towards the pith, and metaxylem, which is formed later in the life, will present towards the periphery. So this is what endarked protoxylem arrangement is known as, and it is common in both monocot and dicot stem. It is the feature for stem. Endarked protoxylem. If we talk about roots, in roots exarch protoxylem is present, which is just reverse of this arrangement. So it is also true for dicot stem. So among 5 features, 4 are true for dicot stem, and it is correctly represented in option 1. Now let's move ahead with the next question. Okay, next question is from biological classification. Again, a kind of rote learning chapter, but again important chapter from NEET point of view. This question is actually asking about the dinoflagellates, the feature for dinoflagellates. Dinoflagellates belongs to kingdom Protista. Protista involve all the unicellular eukaryotic organism. Remember that. This is the feature of Protista. The members which are included in Protista kingdom. Unicellular eukaryotes. That means the organisms which are present in Protista are unicellular and have all the cell organelles which are present in the eukaryotic organism. Now, if we classify them into different classes, it is actually divided into 5 classes, different classes. If we talk about the first one, which is rhizophyte, and first we talk about the habit, then they are found in fresh as well as in marine environment, right? And it include desmids, which is golden algae. This question is also asked that desmid belongs to which kingdom or which group of Protista. Next is diatom, which is very important from exam point of view. The cell wall of diatoms have silica. Its cell wall is actually embedded with silica. Again, very important feature. And the arrangement of its cell wall is like soap box, one over other, like this. If we talk about dinoflagellates, as in the name itself, dino means two. So it is actually biflagellated organism. If we talk about the habitat, it is marine. And they are photosynthetic. In chrysophyte also, these organisms are photosynthetic. That means they have the presence of photosynthetic pigments which can perform photosynthesis. Now, due to the presence of different color compound, they appear yellow, brown, green, red somehow. For example, one very classic example of dinoflagellate, gonolax, which cause red tides in the sea because it appear red in color, right? And if we talk about cell wall, so in the cell wall of dinoflagellate, cellulosic plate is present. Right? So this is what dinoflagellate is. Now let's move to the next, which is euglenoids. Euglenoids are also photosynthetic. If we talk about its habit, it is actually found in fresh, stagnant water. Okay. If we talk about what is the mode of nutrition in them, so it is photosynthetic, it is autotrophic as well as heterotrophic. So how it is actually possible? So when in the presence of sunlight, it will act as a photosynthetic organism, but once it go deep into the water and in the absence of sunlight, it will act as a heterotrophic organism. How? By predating the smaller organism. So this type of mode of nutrition is known as mixotrophic. Again, this term is important from NEET point of view. If we talk about the cell wall, it is devoid of cell wall, first of all, right? And instead of cell wall, it has very flexible pellicle layer, proteinaceous pellicle layer. And it has two flagella, one is very long and another is short. They are not equal in length. Now, if we talk about the slime moles, first of all, they show saprophytic mode of nutrition. Up till now, the organisms are photosynthetic, but in slime moles and protozoa, the mode of nutrition get changed. They become completely heterotrophic. In slime moles, specifically saprophytic. That means they rely on dead and decaying matter. If we talk about its nutrition, so under favorable condition, they form an aggregation which is known as plasmodium. Now this plasmodium will get differentiate and form the fruiting bodies which will form spores on their tip. That means this is one of their strategies to survive in the adverse condition. This plasmodium is actually forming in the favorable condition. In unfavorable condition, what will happening? What is happening? It is forming fruiting bodies. From fruiting bodies, it will form spores which will help in surviving in the adverse condition. This is also very important from the point of view that when plasmodium is formed and when spores are forming. Okay. Now, if we talk about the protozoans, it is basically the primitive relative of animals and they show completely heterotrophic mode of nutrition. If we talk about slime moles, they are connecting link between the plant, fungi and animals. Some features which are equivalent to the plant, some which are equivalent to animals and some are from fungi. So this slime mole is basically the connecting link between plant, fungi and animal And protozoans are primitive relative of animal. Right? And they again are of 4 types. I am writing here. The first one is amoeboid. Amoeboid, as the name itself suggests, that it will be of amoeba-like structure. That means for the capturing of food, it will form the pseudopodia, the false feet. If we talk about the marine form, they have silica deposition. Remember that in the marine form of amoeboid protozoa, they have silica deposition, and in diatoms, they have the silica deposition on the cell wall. Okay? Antamoeba is the example of amoeboid protozoa. Now, next one is flagellated protozoa. As the name suggests, flagellated, that means the locomotory organ is flagella. There are multiple flagella on their body, and the example is trypanosoma. Example is, Okay, sorry for, okay, now it is corrected. So, flagellated protozoa, the example is something has happened. Okay, pen is not selected. Sorry for the inconvenience. Now, let's get back to flagellated protozoa. So, example of flagellated protozoan is trypanosoma. From exam point of view, it is very important because trypanosoma cause sleeping sickness. So, board actually ended here. Please remember trypanosoma, S-O-M-A. Okay, now the next one is amoeboid flagellated. Next one is ciliated protozoa. That means the locomotory organ is cilia. Example is paramecium. Very beautiful feature of paramecium is, or we can say ciliated protozoan is, presence of gullet, where the ingestion of food material will take place. Next one is sporozoan. They have infectious spore-like stage. For example, plasmodium, which causes malaria in humans, right? So, example of sporozoan is plasmodium. So, somehow I try to capture complete Protista kingdom here. This is really important from exam point of view. All these protozoan types and their example, plus the slime molds are also very important. Basically, the complete kingdom, Protista kingdom is important. Now, let's get back to the question. The question is actually asking about the feature for dinoflagellate. Now, let's see the options. The first one is they are eukaryotes. Dinoflagellate, which is present in the kingdom Protista, which includes all the unicellular eukaryotes. So, this option or feature is true for dinoflagellate. Next feature is the cell wall has stiff cellulosic plate. Yes, it is also true for dinoflagellates. Now, if we talk about third option, which says Gonolex is a red dinoflagellate. So, yes, it is also true due to the presence of red pigments in them. Now, if we talk about the fourth option, which says most of them have single flagellum. This option, or we can say the feature, is incorrect for the dinoflagellates, as in the name itself dino is present. That means 2 flagella. And the question is asking about the incorrect statement. So, from this, I can rule out these 3 questions and hit the correct answer, that is option 4. This is how we proceed to the question. Now, let's see next question, which is again from the biological classification. Here 2 statements are given. Statement A is for bacteria, which says bacteria as a group show most extensive metabolic diversity. And next statement is bacteria have simple behavior but very complex structure. So, bacteria is the sole member for kingdom Monera. According to R.H. Whitaker, a very beautiful classification system which is given in the NCERT, which I have taken here. So, if we talk about this 5 kingdom classification, which is basically given by R.H. Whitaker. In Monera, if we see the characteristic, Only Monera cell type is prokaryotes and other 4 kingdoms have eukaryotic type of cell. Right? Remember this feature very clearly. That means the cell type is very simple. No cell organelles, no membrane bound structure, nothing, only nucleoid, cytoplasm, etc. Very simple transcription and translation behavior. Okay? Now, if we talk about the cell wall is non-cellulosic, polysaccharide plus amino acid. In Protista, in the previous question, we have studied that there are certain members which do not have cell wall. And in some members, cell wall is present. I'm so sorry. Now, if we see the fungi, presence of chitin. It is the characteristic feature for the members of fungi that in the cell wall there is deposition of chitin. Now, in the 4th Plantae kingdom, where cell wall is present which have cellulose in them, and in animal cell wall is absent. If you remember, the protozoan, which is the primitive relative of animals, do not have cell wall. Okay? Now, let's see the next one, nuclear membrane, which is absent in Monera. As I've told you that the cell type is prokaryotic. That means nuclear membrane is not present and all the other kingdom have membrane. Next is body organization, where Monera and Protista are cellular, but fungi, Plantae and Animalia, they have multicellular or tissue level of organization. Now, if we talk about mode of nutrition, so among the 5 kingdoms, the most diversity of nutrition we can observe is in kingdom Monera. And the sole member of kingdom Monera is bacteria. Here, both autotrophic and heterotrophic mode of nutrition is observed in the members of kingdom Monera. Autotrophic में chemosynthetic and photosynthetic are given. For chemosynthetic, I am encircling, giving example for chemosynthetic, which is thermoacidophile. If we talk about photosynthetic, classical example, cyanobacteria. And if we talk about the heterotrophic mode of nutrition, it can be observed in the most of the bacteria, for example, lactobacillus. So we can see very diverse way of mode of nutrition in the members of kingdom Monera, and bacteria is sole member. Now let's move ahead in the next kingdom, where Protistans show both autotrophic and heterotrophic mode of nutrition. Where they are photosynthetic, in heterotrophic we have studied that they are saprophytic. Okay? In fungi, they are solely heterotrophic organism. Right? They do not show any any autotrophic mode of nutrition. Okay? And if we talk about the Plantae, here autotrophic, that means photosynthetic. Remember, in the NCERT, it is given that bladderwort and Venus flytrap are insectivorous plant and Cuscuta is a parasitic plant. But this classification system is actually based on the R.H. Whittaker, which is given by R.H. Whittaker. Right? So, if it asked that according to R.H. Whittaker, Plantae kingdom has autotrophic mode of nutrition, so yes, that statement is true. Like that. Okay, so remember all these terms, that this classification system is given by R.H. Whittaker. Now, the 5th kingdom, which is Animalia, it have heterotrophic mode of nutrition, where holozoic or saprophytic mode of nutrition is observed. Right, so this is all the features. Now, question is actually statements for bacteria and statements on bacteria. So the first statement is bacteria as a group show most extensive metabolic diversity. So yes, it is absolutely true. Just now we have studied that it have both autotrophic and heterotrophic mode of nutrition. So from this I can rule out option 4, which is claiming that both the statements are incorrect. Now let's see the next statement, which says bacteria have simple behavior but very complex structure. This statement is incorrect. Why? Because bacteria has very complex behavior but very simple structure. For structure, I have told you that it is prokaryotic, and for behavior, as it have multiple mode of nutrition, so have very complex behavior. So this statement is incorrect. So among the 2 statements, only statement A is correct. Students, keep interacting, ask doubt, whatever you are facing, so that I can clear all your doubts and you can excel in your exam. Okay, let's proceed to the next question, which is from plant growth and development. This question is basically asking about the cytokinin. Cytokinin is the plant growth hormone. If we talk about cytokinin, so there is one scientist known as F. Skook, who was the first one who observed that there is undifferentiated mass of cells which are formed in the tissue culture when he put the coconut milk in the tissue culture along with the auxin. Right? So he basically observed undifferentiated mass of cells. Later on, group of scientists, Miller et al., they autoclaved the substance which have the cell dividing capacity from the autoclaved herring sperm DNA and named that substance as kinetin. They do not occur naturally in plants. That means they are not found in plants. These are synthetic cytokinin. Okay, if we talk about the natural cytokinin, it is zeatin, which is extracted from the corn kernel or unripe maize. These type of questions are also asked. Please don't get confused between the kinetin and zeatin. This is synthetic, this is natural cytokinin. Okay, now if we talk about the site of cytokinin, that where they are produced, so they basically produce at the region where the cell division occur. For example, young stem and roots or fruits. So these are their site. Now, if we talk about the function, the physiological function of cytokinin, so the first function is that they help in the shoot formation or shoot growth. Shoot growth they will help in the lateral shoot formation, adventitious shoot formation, etc. Along with that, chloroplast production is also facilitated by cytokinin and new leaves formation. And it is antagonist to the auxin with respect to the apical dominance. That means it counteracts the apical dominance. Right? What do you understand by apical dominance? That means the apical bud is not allowing the formation of lateral buds. And it also helps in increasing the shelf life of the fruits. Shelf life of fruits. And it also helps in delaying the senescence of the plant. If we talk about the application, so it is used in the tissue culture. If the concentration of auxin and cytokinin are equal, then it will lead to the formation of callus. If the concentration of auxin is more than the cytokinin in the tissue culture, then it will lead to the formation of root. If opposite will occur, the cytokinin is more, then concentration of auxin, then shoot formation will take place. Right? So these are the features of, or we can say the functions of physiological functions of cytokinin. Now let's get back to the question. The question is asking about the select the correct statement with respect to cytokinin. The first feature is it helps to produce new leaves. So yes, it is absolutely true. So here I have to rule out the options which do not contain A. So 3 and 4 get directly ruled out. Next one is it helps to produce chloroplast in leaves. So I have choose the option which contain B. That means option 2 get ruled out. So from this only I can hit the correct answer. But let's see the third feature which says it helps in fruit ripening. It is not facilitated by cytokinin, but it is done by ethylene. So for the cytokinin, only A and B are correct. I hope you understood the concept of cytokinin here, and I hope I have sown the seed of knowledge in your brain. So don't forget to bloom where you are planted. Thank you so much for watching this video.